It's a great. Okay, I'll kick off now. It's a great pleasure to introduce Saul. Saul is a distinguished professor, a leader in NIHR terms, a leader in academic medicine terms, a leader in pediatric infectious diseases, and has been a major leader in the response to COVID-19. But he did comment. That the issue was all these figures from the past. Well, Saul ages me very significantly because, as far as I remember, he graduated before I even moved to the Windair building, and I was in the Windair building for nearly 15 years before I moved to the Cruciform building. So that shows you how that really is a figure for the past, but he was already a prospective professor of immunology and infectious diseases already then. So it's a great pleasure to have him here tonight.
Well, thanks, David. And I can see that you accidentally left the mic on and open to everybody while we were pre-talking, which means I've upset my aunt already.
You haven't upset your aunt, just to reassure you and your aunt that we take the opening conversation until the point that I started to introduce you. The old conversation is deleted from YouTube.
and of course, it is not the public version of the talk. It is the version aimed at doctors. So hopefully that will not put people off. Right, screen share. So, David, can you see that all right?
I can see that fine.
Okay, so David asked me to talk about COVID vaccine trials today. So I'm going to start from the beginning, assuming that there's some GPs and non-infection doctors and some people who haven't been keeping up so much with the medical literature, or in fact even some of the detail in the papers. Newspapers, that is, not the medical papers. So as a very basic reminder, most of you will have seen on the news the pictures of coronavirus and be well aware that this is a virus with spike protein on the outside. And when the coronavirus gets into the body, the cells of the immune system, which are mainly the T and B cells, they work together to produce both antibodies and cells that then remember the coronavirus and respond to future infection, even when the antibody levels have fallen over time since an injection. And then a certain time later in vaccinology, we can use boosters to increase the antibody and help the body remember how to respond. And the body remembers how to respond quickly based on that prime or prime boost, the first 2 doses that we give at the beginning of the course. So what do we need to know about any new vaccine? Well, the first thing is it safe? We need to know if it makes an immune response. Does it protect against infection? We need to know how long the protection lasts. Is there a risk of enhanced disease? Is a special thing we've been looking at for the coronavirus vaccines, because with the original SARS vaccines made 10 to 20 years ago with the first outbreak, in animals with the original vaccines, there was a worse lung disease when animals who had received one of the vaccines then met the coronavirus infection, was challenged with the coronavirus infection. So it's been very important all the way along in all the vaccine trials for us to be monitoring for a risk, or it was a theoretical risk of enhanced disease. And the good news is that we haven't seen any evidence of that in any of the vaccines currently being used against coronavirus in clinical practice or in the ongoing trials. So it is and it remains an important thing we look for in the trials, but it's not something that has happened in humans. We also then need to know about whether we should combine different technologies in sequence. So we currently give vaccines as a homologous prime boost, 2 doses of the same vaccine, and heterologous just means using 2 different technologies, one after the other. And then we need to answer the question, are boosters needed? And if so, when will they be needed and how often? So this is a schematic of the different vaccine technologies that were available towards the end of last year. And the UK Vaccine Task Force, which interestingly was part of the Department of Business, Energy and BEIS rather than Department of Health, invested in 4 what they called buckets of vaccines or different vaccine technologies in order to secure vaccine supply for the UK in what is a post-Brexit world. So the first 2 are not in no particular order. The replication incompetent vector vaccines, so using cold viruses to present the spike protein to the human immune system, are the AstraZeneca, the Oxford AstraZeneca vaccine and the Janssen vaccine. Janssen uses a human adenoviral vector and the AstraZeneca Oxford a chimpanzee viral vector vaccine. The RNA vaccines include Pfizer, Moderna, and a new one that should be available later in the year, CureVac. The recombinant spike protein-based vaccines are Novavax, of which the largest ever vaccine trial in the UK was conducted to prove the efficacy. We're still waiting on the MHRA Reg 174 approval of Novavax, but the data is in the public domain. And Medicago is a clinical trial that will start very soon, is another similar type technology that will be doing a global phase 3, including in younger people in the UK in a few weeks' time. And finally, the Valneva vaccine, which the phase 1 was completed and the results announced a couple of weeks ago, is a different technology. It's an inactivated SARS-CoV virus. That is a very old technology. So like vaccines have been made for many years, grow the virus in cell culture and then inactivate it and use that whole thing to stimulate the immune system. And that might be good because it might be that those sorts of vaccines could bypass some of the problems with mutations. But again, that's hypothetical and not yet evidence-based, either from the assays done in the phase 1 trial, but it will be looked at in the phase 1 and about to be phase 2/3 trial that's going to compare the Valneva vaccine with the AstraZeneca vaccine. So this is the pipeline of COVID vaccines as of this month and the number of doses that have been bought. You'll see, as well as all the vaccines I've just mentioned, the AstraZeneca monoclonal antibody is important, and we'll come back to that later. These are a cocktail of long-acting monoclonal antibodies against COVID that might be better deployed like vaccines, but better deployed in people where the immune systems don't work properly. So how did we set up these studies? So the Oxford team, as you well know, got on with it and developed a vaccine based on their MERS vaccine technology and other work they've been doing for many years. And we've been working with them for a long time on malaria and flu vaccines. So we were the first site outside Oxford to take part with them in the phase 1 this time last year. But it very soon became obvious to the Oxford team that they needed sites, many more sites, and the trial was going to be much bigger. And so this evolved into the large multi-center COV-002 phase 3 Oxford / then AstraZeneca vaccine trial, but it developed into a national network, which is regional based, where the National Institute of Health Research infrastructure, both from the clinical research network, but also from the biomedical research centers and clinical research facilities, came together to create a new infrastructure to deliver large-scale vaccine trials alongside the VTF. And this capacity for large-scale recruitment involves everything you might expect, from research coordination and governance, having to train a whole cohort of new staff, find new places to do the studies. We used a university sports hall for the first 4 or 5 months and then moved into accommodation that was found for us by the NHS elsewhere. It has to be a 24-hour on-call for participants who have symptoms or problems they think might be related to the vaccine. And for the Oxford study, it was the only one to actively monitor subjects for infection. So not just bringing people in for a visit if they experience COVID-like symptoms, which clearly is the main reason for doing the study, to see whether these big phase 3 studies and the vaccines actually prevent COVID infection. but actually to actively monitor subjects, and participants were asked to swab themselves once a week and send that to the national labs. And so the Oxford study is the only one where we've got a definite hard evidence from the clinical trials of whether the vaccines prevent asymptomatic transmission or not. So within my region, Wessex, we realized quite quickly, and many other regions took a similar approach, and some regions needed to, some didn't, but we worked out right sort of in the middle of last summer that we weren't going to be able to do this long term without the NHS and research infrastructure coming together. And actually our integrated care systems in Hampshire and Dorset were amazingly supportive with the logistics, the HR, project management, primary care involvement. And we've established this as an infrastructure where we contract all our studies now through the main trust Southampton, but can deliver the clinical trials across 3 hubs, both in Portsmouth Hospital, Bournemouth Hospital, and our other locations, both in primary and secondary care. So this has given us a really nice infrastructure for the future, where we don't have to contract 5 times in a region for a clinical trial. We've now got the infrastructure and the contracting in place to be able to streamline the setup of public and commercial clinical trials beyond COVID and beyond vaccines in the region, which is a really great legacy. So back to the vaccines and where we are now. So there was much angst and hand-wringing when the JCVI, which is the Joint Committee of Vaccination and Immunisation, made a scientific decision in December to deploy the vaccine in the UK to the 99% of people who were most at risk of death, or known to be at the time the most at risk of death, but more controversially, but with great detail paid to the scientific evidence that was available to them, they deployed the vaccine saying that the second dose should be at 12 weeks, because that would have the best impact on the reduction of hospital admission and death in the UK. It was astounding how people assumed that was a political decision, and it was astounding how many non-vaccinologists decided that they understood the data and evidence better than the JCVI. But that was an amazing decision in retrospect, leading to where we are now with hardly any cases, which almost certainly would not have happened if that hadn't been taken, and if government and JCVI hadn't held firm at that time. So, what else is there about the data from trials that people need to understand? Well, the first is that regardless of the headline figure, 95%, 67%, 76%, whatever it might be, all of the vaccines prevent hospitalization and death for the mutations that are currently circulated. And in fact, the AstraZeneca Oxford vaccine, as I've told you, is the only vaccine so far to demonstrate from clinical trials some effect on the asymptomatic carriage, i.e. prevention of transmission, though, as we now know, especially from data from Israel, data is emerging for Pfizer. It's really hard to gain that data in the real world because you've got different vaccines being used in different populations. But in Israel, where they've just deployed Pfizer, we're getting that data and that's becoming available. And if we ever were or do deploy to schools and secondary schools, there's another opportunity to study asymptomatic transmission in formal cluster randomized trials, which we'll come back to a bit later on. So the main thing you need to know that's not out there is you can't compare trial data directly. Despite what you hear politicians saying, despite what you hear Anthony Fauci saying, or the American members of the FDA, you can't compare all of the trials directly with each other, because regardless of the American claims, which aren't true, the studies did not pick the standard same definitions of COVID when they were done. Yes, now the FDA and EMA, the MHRA, there's an international agreed outcomes for the trials, and everybody's working to the same standards. But the mRNA vaccine trials were done with very strict definitions of COVID. And, just as an aside, everybody's got wound up by this 3-week interval, but there was no basis for the 3 weeks other than it being faster to give. And in fact, most vaccinology would say you're better off leading, even at that point, would have said it's better off giving a longer interval than doing it quickly and then being obsessed by sticking to that 3-week interval. So the AstraZeneca, Oxford and Janssen trials show a lower headline immunity because cases were included with much milder symptoms. So it's like comparing oranges with apples. You can't compare those studies with Pfizer and Moderna directly. What you can do is compare the hospitalization and death rates, which are almost 100% effective in the clinical trials, though the real world is a bit less effective than that, of course, because it always will be. In particular, the adenoviral vectors vaccines are known to build immunity with a longer interval between doses, and Janssen took the view that they were going to use a 2-month gap, but they also did a 1-dose study with over 30,000 people and a 2-dose study with over 30,000 people so that they could generate the data. As you know, the AstraZeneca or the Oxford study was a publicly funded study that evolved in response to public health need and questions that were being raised by the data as it was being generated. And so the protocol wasn't set in stone like most contract commercial clinical trials. AstraZeneca didn't set the protocol. This was an academic-led protocol that evolved. There have now been, I think, 25 or something full substantial amendments as things have changed. But by doing that, it led to very interesting and very important data because there was a variable dose interval. Some of the participants were given doses between a month apart and some much longer, leading to the 3-month gap that we've got between the dosing in the UK now for the NHS. The other thing that was in the news about the AstraZeneca trial was that there was this so-called big error about the dosing, and there was no such error made. The vaccine was being made in Italy, and when the manufacturer was switched to a plant in the UK, it was worked out and established that the doses were being counted in a different way. The concentration of the vaccine was being counted in a different way. And so what ended up as being described as a low dose, high dose in the papers and in the newspapers was actually because of the decision making around which of the formulations and doses to use and proceed with in the different schedules, which again led to very interesting data that eventually turned out showed better immunity because of the length between the doses rather than of the dose used itself. So how does that all impact public confidence? Well, people need to be sure that new vaccines and medicines are safe. And in the UK, we can be completely confident that the MHRA, Medicines and Healthcare Regulatory Agency, do actually have global expertise in medicines regulation. Before Brexit, the MHRA did all of this work for the European Medicines Agency, which was based in London. It was quite a disappointment that politicians and scientists and many doctors made public statements and undermined public confidence without understanding the data or process. From the UK perspective, we've seen that most of the public accept the pandemic vaccines to protect themselves and others, as people do every year for flu. And even the recent rare side effects that have been seen haven't really impacted that public confidence, primarily because the media, with The Guardian probably being the only exception, are being incredibly responsible in the way that they've reported the vaccines. Nobody is scaremongering, apart from 1 or 2 cases. And people have seen the programme proceeding successfully. They've seen their GPs leading it. They've seen they've seen their friends and relatives having the vaccine, and they can be confident that they're doing the right thing. But hesitancy is a problem, is there, it will re-emerge, and oxygen makes that low-level anxiety and overall hesitancy levels worse. There's still quite a high level of concern in the NHS and research communities that the Black and Asian minority groups in the UK and US are still very hesitant, driven by social media and misinformation in closed groups within their communities. And somehow we need to keep this clear, transparent communication from the JCVI, the NHS and government. You know, we could have a long and interesting debate about whether governments globally should regulate social media misinformation. They could do it, they could clamp down on it. They've still chosen not to do that. They've labeled it, which probably isn't good enough. The other things that we could do, such as giving benefits for people who've been immunized, that has been pretty successful in Israel, whether it's to do with being able to go places or do things. These are much more complicated in our democracy and have really high potential to do harm as well as good. In particular, the UK public will probably perceive this as compulsion if we were to do that. And that's sort of been the political debate at the moment. That's likely to be detrimental to public opinion and drive hesitancy, even if it looks like we're going to need these so-called passports for foreign travel. Now, that's everybody, you as well as I, can have just opinions on sort of internal vaccine passports and the need for the foreign travel. But for medics and hospitals and healthcare workers and social care workers, immunization is a specific category that maybe should be treated as a special case. It would still need considerable consultation and workforce support. It is being considered for care homes, but why don't we have compulsory flu and COVID immunization for us working in healthcare? Everybody accepts that you go to occupational health when you start a job and you have to have hepatitis B and chickenpox vaccines to work in hospital. It's sort of a mystery to me why we haven't done that for flu years ago and why we don't do it for flu and COVID now. So what do we know about the vaccine effectiveness in the real world? Well, I'm not going to go through the data and the many, many papers and articles now, but there's good data from Israel, Scotland, Public Health England, the United States now is not on the slide, but there's a particular study that was done in Bristol by my colleague Adam Finn and his colleagues that was released by preprint on the 3rd of March. It's not yet published, but it's worth just showing you, because this, if you like, is the justification for that JCVI decision. So the AvenCap study was actually set up last summer. It was funded by Pfizer as a surveillance project in Bristol's 2 large NHS hospitals, where they recorded detailed information on every adult patient admitted with symptom signs and X-ray evidence of acute lung disease. And they estimated the effectiveness of 1 dose of the vaccines by taking the acute respiratory disease cases who'd had a positive test for COVID on a hospital admission and those whose test was negative, and by comparing the immunization rates in the 2 groups. They structured the analysis to make comparisons within individual weeks to minimize the biases due to the rapid changes in the epidemic and the vaccine rollout. And they've controlled for social deprivation, home care residents, sex, and other factors. This was amazing data, because what it showed was that 1 dose of AstraZeneca was actually a bit better than 1 dose of Pfizer. But the most important thing was that they were identical if you took into account the time period and reconciled the data. 79.3% for Pfizer and 80.4% in the AstraZeneca. In patients, look at the middle of the slide, with a median age of 87 and 88 years respectively. This is an unbelievable, for those people who work in vaccinology, this is an absolutely unbelievable real world effectiveness, hard data, because we just don't normally see vaccines to be that effective in people of this age with immune systems that are normally much less responsive to vaccines. So this is just fantastic for society and for the deployment, and is the reason why our hospitals are now not overwhelmed and intensive care units are not full of people dying from COVID. So what about the very rare side effects and other real world data? Well, I'm not going to go into the detail and the detailed biology. I don't have time and I'm not a hematologist, but the extremely rare blood clots seen in AstraZeneca and the Janssen vaccines when deployed to millions of people are not yet fully understood. It could be the vaccine type, i.e. the class, the fact that they are similar class vaccines. It might be due to antibodies to the spike protein, or it could still be something else entirely. What is clear is that we absolutely need active surveillance for all new medicines and vaccines, and that includes every new vaccine, whether it's mRNA or viral replication incompetent vector vaccines, or whatever it might be. We have to be cautious about data being reported. you've seen a few and a handful of cases from Janssen from the United States deployment. The US has notoriously bad surveillance systems, despite so-called enhanced surveillance for COVID. And so we have to really wait, although Janssen's now reapproved and being reused in the over 18 population in the United States. It's now started its deployment in Europe, and it's only now with European surveillance systems that we will properly understand whether it becomes a class effect and Janssen AstraZeneca behaves similarly in people in the population when the vaccines are deployed, or whether Janssen and AstraZeneca behave differently with regard to how rare these side effects are. But surveillance is absolutely key. So I'm not going to go into these in detail. There's some brilliant public risk and evidence communication pictures that were in the papers that were developed by the Winton Centre for Risk and Evidence Communication and used by Jonathan Van Tam in his 5 o'clock briefing a couple of weeks ago. I've copied the panel from the BBC on the left, so you can see there's some, really nice information that can be shown to public and patients who are worried about the relative risks of the serious harm due to vaccine side effects. Even if you're 25-year-old, you've got way less chance of having a harmful side effect from a vaccine as you have of dying in a road accident or dying of coronavirus or being hit by lightning. And the complicated panels on the right, which you can sort of show in a webinar where you're all on your computers, but you couldn't put up in a lecture room, just shows you the reason for the decision to not use AstraZeneca in the UK in people under 30. That wasn't a regulatory decision. The MHRA have not put any age restrictions on the AstraZeneca vaccine, and the FDA and EMA have not put any age restrictions on Janssen, which isn't yet approved for use under Reg 174 in the UK. But you can see it's just worth looking at these are the older people. Here you can see the low exposure risk. So this is where you've got low COVID in the community, top, medium COVID in the community, middle, and high COVID immunity in the risk in the bottom. So high transmission, like in January. And you can see that for all ages, except the 20 to 29-year-olds right at the top here, the risk of ending up on ITU due to COVID is far higher than on the right in orange, the risk of a harm from the vaccine. But you can see, if you look at the young people, the risk of ending up on ITU is still higher here for the COVID on the left and the vaccine on the right, where there's medium COVID, and quite high if there's a lot of COVID. But when we move into low transmission, which is sort of now in society, the relative risk of the vaccine due to this rare side effect and COVID is about the same. And that's why for the under 30s, the JCVI have recommended we use a different vaccine because we have them available. But if we didn't have them available, JCVI would have said carry on using AstraZeneca. But we have options. I've just said this. The detail is there on the JCVI website and was updated again today on the Public Health England website to reassure people that it's a JCVI decision. We're not using AstraZeneca in the under 30s because there are alternatives, but we should be carrying on and giving people their second dose, because at the moment, even there's no signal, even in the under 30s who've been at risk, who've received their first dose of AstraZeneca. At the moment, there's no signal from the second doses, but that is under surveillance. And today there's an update that some of you will have seen on the PHE website, putting the risks and benefits even clearer to do with COVID and not COVID. And I'm not going to go through this slide. This was just a signpost to you that it's been updated today on the PHE website for you to go and have a look at. So what do we need to do now for the last section of the talk? Well, we've still got the Vaccine Task Force supported phase 3 trials. Valneva, I mentioned earlier, is going to be just a UK study. This is a vaccine made in the UK, whole viral vaccine, and it's going to be compared to AstraZeneca Oxford. It's got a bit complicated in recent weeks because the under 30s are only going to, they're going to be randomized to only receive the active vaccine, whereas the over 30s are going to, and that's going to be clearly won't be blinded, but the over 30s will be blinded, so there's a blinded comparator. Medicago is going to be a placebo blind trial globally, not just in the UK, but there'll be a small cohort of the younger age group of people in Britain who still haven't received NHS vaccine. We still have an ongoing need to look after all our trial participants from all the studies we've been mentioned, whether it's the Janssen 2-dose study or Novavax or the Oxford studies, or the Valneva studies and so on. The NHS deployment has been interesting. We now have crossover studies for Novavax and for Janssen, where people who had received placebo have either, for the Novavax study, people had received 2 doses of placebo, received 2 doses of the Novavax vaccine and vice versa. And Janssen handled it differently because their 1-dose study has been approved for use in Europe and the States. The placebo group in Janssen are crossing over to a single-dose cohort, and the 2-dose cohort are staying as a 2-dose cohort, and that unblinds rather than remaining blind like Novavax is doing. We have ongoing safety and immunology follow-up for all the trials, and all our participants are really anxious about vaccine passports, and there's a piece of work going on for all the vaccine trials together between NIHR and Department of Health and policy team and NHS England to make sure that there's no disadvantage to people who've been on trials if an internal vaccine passport scheme was introduced, and a separate conversation about what to do for people going abroad, if in fact we are allowed abroad with all the mutations and third wave happening around Europe at the moment. But that is all unknown at the moment and to follow. This summer, we've got a series of trials led by the National Vaccine Immunisation Evaluation Consortium, funded by Vaccine Task Force, CEPI, and Department of Health. The COMCOV study is a scheduling study looking at whether we can use one vaccine and then another, the so-called heterologous prime boost. And COMCOV2 is now a similar, identical protocol. So this is looking at AstraZeneca, Pfizer, AstraZeneca, and Pfizer compared both at 4 weeks and 12-week intervals to generate the immunological data we need to make decisions in the future. And CEPI have put funding into that because it's globally really important. With vaccine supply issues across the world, people need to know whether if they run out of vaccine after giving 1 dose, they could use another one for a different dose. COMCOV, it's not going to be so useful for the UK, but might be this summer if we run into supply problems. COMFLUCOV is a study being, so COMCOV and COMCOV2 are being led by Oxford. COMFLUCOV is being led by Bristol, and that's asking the question, can we give one of the flu vaccines, and of course we give different flu vaccines to different age groups in the NHS programme, so all of them are being tested, at the same time as people are receiving their 2nd dose of Pfizer or AstraZeneca. That means 1 vaccine in each arm, not at the same time, not combined in the same vial. That's absolutely critical for the NHS for the autumn, because unless we can give boosters at the same visit, there will be complete chaos for our general practice and NHS vaccine deployment colleagues. And the COV Boost study is going to ask the question, can we safely use a booster of one of the UK available vaccines? The Boost study we're leading in Southampton. It's going to be an 18-centre study. We're going to have 9 investigational products. So we'll have 6 COVID vaccines from the ones I've shown you, and 3 of them will also be being tested at a half dose. And that will report to JCVI by the end of August to inform the JCVI decision about who and which boosters should be able to be used in the autumn. So at the moment, it looks like we're gearing up for JCVI to be able to make a decision on whether people should receive boosters in the autumn. The decisions will be made not just on the basis of these 2 or 3 trials, but also the longer term immunity data being generated from the original Oxford and Pfizer and Moderna and all the other trials that have been running globally. that is available to the vaccine committees, albeit not all of that data is necessarily in the public domain. So we would expect to see a booster in some people in the UK in the autumn, but whether that will be everybody or just the most at-risk groups, we do not yet know. There's also studies in pregnancy, children, and immunosuppressed people, and I'll just show you a little bit about them. So I've told you about ComCov, don't need to go into the detail just now. I've told you about ComFluCov, and I've told you about Cov Boost. So for pregnancy and children, these are becoming a bit more complicated because of the decisions both by the JCVI to offer immunization now with Pfizer to all pregnant women who want it, but also because of the pauses to the Janssen and AstraZeneca vaccines deployment for the different reasons at different times in different domains around the world. So we were about to have all of the three of these vaccines enter trials in pregnancy in the UK. Pfizer's going to happen. AstraZeneca's on pause because it's not being used in trials in the under 30s. And Janssen, we do not yet know because the company haven't decided what to do with their program. Similarly for children, the Oxford vaccine study is on voluntary pause. The sponsor has put the study on pause pending further information. And the MHRA have agreed with that pause. And again, the Janssen teenage and children's studies in the UK and globally are on pause pending further decision making, and may well be that both of these studies, both of these companies' vaccines are some time before they start again, before we understand the clot risk, because children don't get severe COVID. They might not even get bad long COVID in the amount that some people in the media are saying. And it's really important that we wait for the UK clock study being led by Terence Stevenson to tell us the long COVID rates in children, because of course many of the symptoms of long COVID in children, depression and so on, might just be a reflection of lockdown and being people not having their children not having their normal lives. So the clock study is looking at a very large cohort of children who've had COVID tests, some of whom are positive, some of whom are negative, and that will report next year. In the meantime, you know, the multi-system inflammatory syndrome is incredibly rare and seems to have gone away now. So if we deploy vaccine to children, it will be the only reason we can justify for being for children's benefit is because it will keep schools open. And actually, we might be able to keep schools open by immunizing the adults and keeping COVID rates low in the community. So vaccines in children are not going to be deployed soon. If there were a sudden wave of a really transmissible variant that JCVI felt that immunizing with Pfizer would be useful for, because it's only Pfizer we can use for the kids, and even then the data in the States is only for the 12 to 15-year-olds, they might do so, but they won't deploy to children without the relevant safety and efficacy data and children being included on a Reg 174, and that isn't going to happen soon for anything other than Pfizer 12 to 15-year-olds in the UK. Even if we wanted to deploy to children, we still have to balance the vaccine supply for those vaccines with what we need to do for the adult and at-risk population. So there's no certainty that even if there were a decision to immunize children, we would have enough vaccine to be able to do so. So what we're not going to do is vaccinate children so people can go on holiday. That is not the way you manage public health in a pandemic. So for immunosuppressed people, monoclonal antibody might be a way forward. So these are very long-acting monoclonal antibodies. AstraZeneca is the one we've trialed in the UK. Provent and Storm Chaser, we're waiting for the primary outcome results to be announced, a bit like the vaccine ones were. These are attractive. The technology is not new. We've been looking at these in RSV and other respiratory viruses for children for some time now. And palivizumab is obviously given to preterm infants every month. These are next generation monoclonals with very long half-lives. They last 6 months. And just like the RSV 6-month monoclonals, which are in the late stage clinical trials now for RSV, these might well be a good way to protect people with cancer, a shot every 6 months, say, or other immune conditions if people don't respond to vaccine properly. And the Octave trial, led by colleagues in Birmingham, is investigating the effectiveness of the COVID vaccines being used in the UK in 4 specific conditions: cancer, inflammatory arthritis, diseases of the kidney and liver, and having a stem cell transplant. So from what we know of antibody level studies being put in the public domain, it might well be that the vaccines are not so good at people whose immune systems don't work. That won't be rocket science as a result, but it could be that monoclonal antibodies provide the solution for those groups, or it could be that using different dose strategies is the right thing to do. But we've got quite a lot more work to do before we understand that properly. Adults who are shielding obviously are much more vulnerable than children. One thing is clear is that the recent press, again, that Radio 4 have done some work, did some pieces on this over the weekend, but there appear to be 40 or 50,000 children shielding unnecessarily, whose specialists have not been asked to advise, and parents have been receiving text messages from GPs incorrectly from NHS England telling them to shield, and have been made very anxious. But the one thing the paediatric community is pretty clear on is there are minuscule numbers of children who actually should be shielding. And despite the NHS England mistakes, we need to be empowering parents to get those kids back to school without vaccine. So the live viral challenge trials might help us accelerate vaccine development. There's extensive UK experience for bacterial and parasitic diseases in Southampton, Oxford, London, Liverpool, and there's a UK collaboration involving multiple institutions led by Imperial College. We did the detailed public involvement work to demonstrate the feasibility in Southampton. These are studies where you invite young, healthy people with no COVID risk factors to come and receive new vaccines and then be given COVID, or some of them get the vaccine, some of them don't, and you give people COVID to study their immunology and see whether the next gen vaccines work better. And of course, this might be a good way of testing vaccines against the variants as well. It's quite complicated ethics because, of course, we've got no treatment for viruses and for COVID as we have for bacterial and parasitic diseases when we do this sort of work. So people have to go in with their eyes open, but it's not for doctors to decide those ethics. It's for us and researchers to explain the risks and benefits to people who might want to take part, and for the participants to fully understand all the risks and possible benefits to society, if not themselves, and look at what financial incentive they might be being offered, and they can make a decision. We don't tell people they can't be North Sea oil rig divers because it's a high risk occupation. We just pay them hundreds of thousands of pounds a year to do it. And these trials work on roughly the same ethical principles. The obligation is for researchers to give that information and everything in an understandable way to potential participants who are able to then make up their own mind as to whether to take part. So finally, in summary, what are the key issues for the NHS in the autumn? Will a third dose booster be required? Which vaccines are we going to use? Different technologies or the same? Should we use Wuhan strain or variant strain vaccines? There aren't any variant strain vaccines available yet, but they're just starting to come through to clinical trials, and COV Boost will have another stage later in the autumn where any UK available variant vaccines will also be tested in the same protocol. We don't know yet whether it's going to be all ages or just over 50s, or just an even smaller group. We don't know about whether we're going to be giving flu at the same time, but we hope we'll be able to, and maybe we'll be able to do studies looking at the prevention of transmission as we deploy to teenagers and or children, maybe in 2022 or even later. But I guess we probably will be looking at vaccine in teenagers next year is the best guess at the moment. So hopefully I've shown you where we are at the moment. We have got an ongoing need for clinical trials of different vaccines and technologies to ensure the global supply. We've still got a lot of work to do around optimizing protection, looking at vaccine combinations, different ages, different clinical conditions, and addressing issues caused by virus mutations. There's issues with the global manufacturing supply chain that will impact on how we use vaccines. And we have to somehow avoid this absolute nonsense of vaccine nationalism and people doing things like the EMA taking AstraZeneca to court to try to make themselves avoid the fact that they hadn't done their contracting and due diligence properly, and to deflect what was pretty shocking politics all round. You know, sue the drug company instead of address your own problems. But somehow we've got to avoid that vaccine nationalism. So thank you for listening, and I guess there'll be a few questions that we may have time to answer. David.
Well, thank you very much for a wide-ranging survey of a very interesting, topical, and exciting subject. There are a couple of questions which were submitted before and are submitted now, and far away you mentioned the question of Newton's in your last slides, the question about are you worried about the question of viruses evading vaccines, particularly?
Yeah, I think people are worried, but I don't think it's as bad as people are worrying in the press, and the answers aren't going to be immediate. So we know we can bypass mutations using vaccines by doing one of two things. One is to increase the antibody level generated from the Wuhan variant, and the South African data, particularly for Janssen, has shown that you can avoid hospitalization and death by increasing, having high antibody levels. The Novavax trial similarly in South Africa showed that. And the other way is by using a variant vaccine. Now, the problem with variant vaccines is you have to trial them. You can't just assume that using a variant booster is going to work. There's a immunological phenomenon called original antigenic sin, which sort of has a theory, and David, you'll probably be able to tell people how this works better than me at this time of night after a long day. But where in hamsters there's some data and other animals that if you use a variant vaccine after using the other one, the original strain, you actually get worse responses against the variant than you do against than you would if you used a booster of the Wuhan variant in the same group of animals against the variant vaccine. So against the variant strain. So it can make the easy way of sort of describing that is using a variant vaccine can make your immune responses to the variant worse than if you use the wild type vaccine. So we have to do the trials, and we have to look at how the assays in the lab for the trials are being done against all the major strains. So the booster studies we're going to do in the UK will test both antibody, but also the pseudo-neutralizing antibodies and the neutralizing antibody assays against the important strains that have been decided that are important for the Vaccine Task Force studies in the UK. And the labs for all of these studies are quite centralized, both at Public Health England and specific laboratories around the world, so that the comparators, the appropriate comparators, can be made. So we're pretty confident that with the current modeling, we are going to be able to avoid the problem of vaccine escape in the at-risk populations by using a booster in the autumn. Though what that booster will be, we don't yet know what's going to be the safest in terms of the side effects. So we need to boost looking at, what if you had a third dose of Pfizer, would it just be too painful or give you too much fever? Or if you use a third dose of something different, would do the immune system goes up and down across all the vaccines that will be available? So we will be prepared with the data from the UK, and the companies are having to do their regulatory studies for the variant vaccine, so both in naive individuals, so using the variant vaccines from the start, and as booster doses. So there will be data, but it won't be there till early, mid to late autumn about the variant vaccines.
Right. Okay. There's a number of questions that relate to interference and other medications. One of them is about biologicals, the people on rheumatoid arthritis, inflammatory bowel disease getting biological therapy. And what about the question about interference between the different vaccines? Is it safe to give your hepatitis or your pertussis or your pneumococcal at the same time as you're getting the coronavirus? Is there any cross interference between?
So for all vaccines, we normally give a month between doses of different vaccines, especially the live ones. the non-live vaccines have traditionally been able to be given at the same time, but only after clinical trials, right? Which is what we're doing with the ConFlu-Cov study, looking at if we can give flu and COVID together. So the advice is for people to wait 4 weeks between a dose of any vaccine and COVID vaccines at the moment, because the data is not there. But in 3 or 4, it's amazing, isn't it? I read something just before we started this talk that it's something like we just passed the billionth dose of COVID vaccine given in the world 5 months after the first approval. Everybody wants the answers to the long-term what if questions, but the fact is that in November we didn't have any COVID vaccines, and we weren't even sure that any of the vaccines would be effective. So this is truly astounding science. But how we're going to need to, if people are going to need boosters every year or every 2 years, or with variant vaccines, or how to use vaccines in all of these groups, whether you've got biologicals or cancer chemotherapy, are all studies that need to be done and won't be quick. So we need to know whether things like the monoclonals are sensible to use in the meantime, and actually preventing COVID in the wider population by immunizing everybody is a good way to start.
Yeah, I mean, there are also the questions that are being asked about the monoclonal antibodies. Is could it be the monoclonal antibodies, passive immunity a la Donald Trump is presumably no longer a serious possibility.
Those are the AstraZeneca ones. So the Provent study is using it as a prophylactic. So using the monoclonal like a vaccine, give it before any sign of infection. Storm Chaser was a similar protocol, but was giving the monoclonal to people who'd been in contact with a positive case, but who hadn't yet got symptoms. And we don't know the results of those trials yet. But based on the RSV clinical trials using similar technology, we've got reason to be hopeful that if immunization proves not successful, if active immunization proves not successful in certain patient groups, that we'll have some alternative.
Right. Okay, so now we've got the question of the allergy. Incidentally, it's a question I've been asked many times. Is the liposome the cause of the allergic effect?
To be honest, David, I think you should take the answer to that question. I've not been so involved, and I gave up allergy a long time ago. I also learned quite early on in the pandemic to keep away from my non-specialist subjects. So I'll let you talk to allergy.
No. Well, I'll find. I'll pass it on to Benny, because he actually did use liposomes at one point. But there are the question about what are the surrogate markers that can be used, the surrogates that can be used. Maybe you could comment.
Yeah, so we don't yet have the surrogate markers of infection. Whether it's going to be spike antibody levels, or we can correlate that in healthy individuals. Of course, it won't be the same for people with cancer on biologics, with a pseudo-neutralizing assay or with actually the full neutralizing assay. All of this is to come, because the neutralizing assays, especially to the different variants, are really complicated to do. In fact, I don't think it would be wrong to say that PHE Portadown are one of the only places in the world that are doing these consistently, and they're doing them for many of the company of almost all of the global trials. That's the full neutralizing assay. This is complicated viral virology and vaccine assays. So what people is probably useful is what you can't do is use a commercial spike assay to decide whether you're protected or not. It's really odd what Israel are doing. Test when you go in, and you get in if you've got antibody, because of course the commercial assays are simply measuring what will have been validated to prove COVID disease or not. They're not valid. And in a vaccine trial, you get a whole range of antibody levels, as Liz has indicated on the Q&A.
Yeah.
If some people don't respond, but almost certainly they're going to have some degree of T and B cell response, and they might have cellular immunity, and they might actually be protected. We just can't prove it with a lab assay. So I think one of the things that from the vaccine research community we've been clear on is people shouldn't be doing tests to see whether they're protected, because you just won't know whether if you're positive, it might not mean you're protected, and if you're negative, it doesn't mean you're not protected. And actually, the surrogate markers of infection will be important, and there will be a global standard that WHO and CEPI approve, but not yet.
There's another question which we can't answer, is it the anti-PF4 antibody for the thrombotic complications or cross-reactive COVID antibody? I don't think,
no, but great science, great science to have got those, to have made that observation, to have published it, to be put it out there. And, and people saw these side effects as a problem. They're not a problem. It shows you what fantastic surveillance we've got, both in Europe and in the UK. The problem has been the politicization of what is an incredibly rare side effect that needs reporting and monitoring, but not scaremongering with.
I mean, the question about the wait, the delay in giving the boost, the delay after having the monoclonal antibody therapy, and indeed the delay that the question again that a lot of doctors ask me is, if I've had a COVID-19 infection, isn't one shot enough? Because isn't one shot a booster for me rather than a...
Well, that depends on that. I mean, and again, we will get some information by using the heterologous boosting and heterologous. We just don't know enough about the immunology of coronavirus infections. We still don't know why children don't get severely affected, for goodness sake, and that might also give us clues as we understand that better in the future as well. So they're all great questions, but not ones we're going to answer tonight.
Yeah, and they're going to take some time, and you've answered quite clearly that the booster data is going to come out towards the end of the year.
But we'll get information on how boosters respond in terms of being able to exclude them from the use in the UK program. So if you think about what's possible and what's not possible now, right? If we enroll people to the booster study in June, they will only be 3 months away from their last vaccine. So that isn't even real world. They've only had they've had 2 doses, 3 months, 3 months apart, and then they're going to have another 3 months and a third dose. So the booster trials this summer are going to tell us whether it's safe to use the vaccines as boosters this autumn, i.e., do people get too many side effects? And are there any of the vaccines that don't show boost against control? What they're not going to tell us is how long immunity lasts or whether we need boosters every year. We're going to have to look at the long-term follow-up from people on the COV-002 study, the long-term Janssen and Pfizer studies, people who take part in the COV-Boost study this summer will be followed for a year. And by next summer, we'll have a bit more information about how long immunity lasts. And we'll also have, as well as the people in the vaccine trials and the booster trials, we'll have people who don't receive a booster this autumn, but receive 2 doses this summer, and we'll be able to measure their antibody levels next summer and know what's happened with only 2 doses and a year. So there's an awful lot of work to do, but an awful lot of answers that can't be given for some time. And you've frozen, David. I don't know whether I've frozen, whether everybody's frozen. You froze there, David, or did I freeze?
That's better.
Did you freeze or did I freeze?
Both of us did, I think, because it said my connection is unstable, but it was the connection, maybe the connection to you. So John Posner raised as a clinical pharmacologist, raised the question of the fact of who decides on risk, that the doctors decide on risk, and you said the patients decide on.
Well, I think for the challenge studies, it's the researcher's responsibility to present all the information to the ethics committee, who then decide whether it is enough information and presented in the right way. It's then up to an individual whether the risk benefit for them is right for them as an individual. So society has to decide that we're going to do that sort of work. And then, just like deep sea diving, it's an individual choice whether you want to do it or not. What we can't do is coerce people into any form of clinical trial.
Right.
So I don't know whether I described that wrong, John, to your question. I think it's a researcher's responsibility to present all the information, but ultimately it's an individual's decision whether to take part in a ethically approved clinical trial.
Right. Then just the one thing we missed out on. Liz suggested that with the rituximab, you should give Pfizer, not AstraZeneca. I mean, has there been any? I don't think there's any evidence.
Hopefully, the communities of really engaged clinicians and specialty groups in the UK who have been collecting samples as their patients have been immunized, hopefully those questions will have answers soon as well, because there's some really good groups who've been coming together. You know, all the different cancer biologists have come together doing, I think there's 8 or 9 different studies being done around the country out of, you know, people just getting on with it. And, you know, Cancer Research UK have put out a call for a meta-analysis already of all the data that is becoming available. So, you know, all the work the specialists, the subspecialists are doing in the UK, gathering the samples and collecting the clinical information is going to be critical.
I think that, you know, we now past the official hour of 9 o'clock. And I think I want to thank you very much for an absolutely fascinating and very well explained talk. We need to actually consider, when I was reflecting on what you were saying, I was thinking that you were also posing quite a lot of questions for us.
For everybody, yeah, for the world. And I think we have to have patience, don't we? We have to do these studies properly and not over-interpret or interpret wrongly. There's a lot of non-expert experts on the news all the time at the moment.
Right, and well, you've certainly shown us your expertise in a very remarkable way, and of course, it did make me think back that yes, I do remember that you were always very good at asking the questions all those years ago. So thank you very much, and thank you for keeping on asking all these questions.
No problem. Thanks for inviting me.
Thank you.